in order to understand meiosis, which is what our next project is, you have to remember the difference and understand the difference between homologous chromosomes, sister chromatids, and just different chromosomes. So you need to understand what it means to be a different chromosome, you need to understand what it means to be a homologous chromosome, and you need to understand what it means to be a sister chromatid. So what we're going to talk about is the chromosomal basis for all of the patterns of inheritance that we're going to learn about in the next section of material. So if you understand this stuff, you will understand the laws governing inheritance, because they're all derived from what happens during meiosis. So you've got your homologous chromosomes, and how would you describe homologous chromosomes?
They're two chromosomes, like one's from your mom, one's from your dad. They're very, very similar, but
So it's the chromosomes that you get, the two versions of a given chromosome that you inherit from mom and from dad. They contain exactly the same series of genes on them, but they differ in that occasionally one will have a different allele than the other. So they're really similar, 99.9% similar, right? And that difference is what underlies the difference between any two individuals here. So when it comes time to copy a cell, the first thing you have to do is you have to copy the DNA. So we sometimes refer to this as replication. That is, you're replicating the DNA, copying the DNA, or synthesizing DNA. So those terms are sometimes used interchangeably by me, and I should stick to one, but I think I just naturally use them differently, or to mean the same thing in different circumstances. So don't get confused. If we say synthesis, replication, DNA copying, all we're talking about is the splitting of the double helix and the synthesis of the new complementary strand. That's all DNA replication is. So when you replicate the DNA, you split this double helix, you synthesize a new copy, and now you have sister chromatids. And sister chromatids, of course, are 100% identical, barring any errors that happen during replication. Okay? So when any question on a lab, or probably we wouldn't give you this on an exam, we'd make it clearer, but if any question asks you to sketch a cell before S, that is before the DNA synthesis phase of mitosis, excuse me, you'd be sketching it before you've made the sister chromatids. All right? If you sketch after DNA synthesis, you would have sister chromatids present. So that's just one thing to keep track of in terms of some terminology that still kind of creeps into the laboratory despite our best efforts to eradicate it. All right, so let's watch this video just to sort of cement it all in place. So that continuous process, we were able to capture in a still image by taking root tips, sectioning them, staining them, and fixing them onto a slide. And so you were able to look at some of those images and find cells that they're permanently arrested because they're dead in different phases of mitosis. But remember that the process of mitosis is a continuous one. They don't do one, stop, do the next. It all flows in one to the other. So when you look at this, just remember, this is anaphase and metaphase, anaphase. So you can capture them at different points depending on when you actually kill and section the root tip. So this is what you guys were looking at. So are you guys ready to talk about meiosis? Yes?
Yes.
All right. So in meiosis, there's several important differences with mitosis. The first one we'll start with is the fact that meiosis only happens in the cells that give rise to eggs or sperm. So meiosis only occurs to give rise to eggs or sperm. And the generic term we use for eggs or sperm is gametes. Meiosis is not a process that only involves sex chromosomes, and sex chromosomes don't only go through meiosis. All right, we'll have an activity you guys sketch out today to try to confirm that. But don't get confused that by the fact that we call certain chromosomes sex chromosomes, that there's something different about them in terms of meiosis or mitosis. They're present in all the cells of your body, and they go through mitosis and meiosis. So something else different about meiosis is that sometimes its name, let me rephrase that, the word meiosis can be described as reduction, division, because what happens in reduction division is you actually reduce the amount of chromosomes in the cell. So what you do in reduction division is you take a 2N or diploid cell, and from that you make N or haploid cells. So you reduce the total amount of DNA that's present in the cells. And when we did mitosis, was there a reduction in the amount of DNA between the starting cell and the daughter cells? They were 100% identical, right? 100% identical. So another part of this reduction division is that unlike mitosis, where you go from one diploid cell to two diploid cells, in reduction division, or meiosis, you go from one diploid cell to 4 haploid cells. So what we'll do in this is we'll go from one diploid cell to 4 haploid cells.